Elrod, August 5th, 2025. Cells, prokaryotic, eukaryotic, their interaction. All cells, all living things are composed of cells. The cell is a basic functional unit of life, and cells, they arise from pre-existing cells and carry genetic information. Our first cell is a prokaryotic, which is in the bacteria and archaea domains. This is a picture from the Campbell book, which is a bacteria domain. We have the outer region here, which is capsule, cell wall, membrane. Capsule for protection, cell wall for structure. Membrane is a in and out barrier for things that go in and out. Flagella and fimbriae. Fimbriae for attachment, flagella for movement. Then we have our inner region, which is a nucleoid with chromosomes and some ribosomes in cytoplasm. So the ribosome is very important to synthesize the protein, and the nucleoid region has the DNA DNA structure instructions for the cells to for the ribosome to carry out. Eukaryotic cell. I did a similar thing here I did with biorender. Basically, I got a famous picture from Johns Hopkins and I labeled it with biorender, similar to what I did with prokaryotes. This is Animalia Kingdom. There's the kingdoms, there's a few of them, and they kind of depend on what the food source is. So the Animalia, which is our cells, the food source is other organisms. So there's I knew it was Animalia because there's a lack of a cell wall, and there's a lot of like internal partition membranes, so it's compartmentalized. And again, that's structure. So the compartmentalization, all these things that I labeled, that's going to be lead to more function. Which one of the functions is genetic control. Over here we have an envelope, the nucleus with the chromatome, which has the DNA, the replicating genetic instructions for making the RNA. And mRNA over here is going to get shipped into the cytoplasm. We have your nucleolus here. That's going to actually create rRNA, which is going to assist the ribosomes in turning mRNA into proteins. rRNA is like ribosomal instructions. So another area of the function is going to be your energy input mitochondria. Again, you have some structure like folding in the mitochondria. The more folding, the more ATP can be produced for energy. That's your energy processing function. You have manufacturing, distribution, and breakdown over here with your Golgi, your ER, your mRNA, ribosomes, vesiculas. Ribosomes produce the proteins that continue to grow in the ER and the Golgi. The rough ER has the ribosomes wrapped around it. It's going to make some molecules and proteins. Smooth ER is going to make lipids, do some detoxification, and your vesicules are going to actually be shipped out from the Golgi. One thing would be that actually gets shipped are lysosomes. Lysosomes are enzymes that one thing they do is break down mitochondria when it's not needed. So we have other function area like structure, movement, and skeleton. Here we have cytoskeleton. It's a really cool video on a protein walking on a cytoskeleton in the class notes. So that's your movement, structure, communication, all in one. You have your membranes here with your lipids. That's going to be for shipping things in and out of the cell. One way I was able to distinguish between like the mitochondria and the ribosomes was there's a lot more, like millions of ribosomes, like 100 mitochondria. Here's a lot of differences and several differences that I pretty much went over, some similarities. Now we're going to go into interaction between prokaryotic and eukaryotic cells. And I have a few examples, but it's going to all start with the making the prokaryotes make short chain fatty acids. And short chain fatty acids actually signal to the eukaryotes. And the way it makes it is it's like makes some enzymes that cuts down to polysaccharides, monosaccharides, then go back into the gut bacteria and they ferment into a pyruvate. The pyruvate goes into the metabolic pathways and creates a short chain fatty acid. And this short chain fatty acid is a signal for the receptor. So when the receptors get stimulated here, these receptors are going to actually open up pathways in the vagus nerve that actually facilitate the transmittance of serotonin, which is a molecule, which is a neurotransmitter to a neuron, a eukaryotic cell. Serotonin is produced by entotrophin, another eukaryotic cell. So the eukaryotic cells are communicating by neurotransmitters. The entotrophin is in the stomach, and the prokaryotic is facilitating the communication by helping to open up the pathway on the vagus nerve. That's big interaction one. Big interaction two is adipose tissue. Your short chain fatty acid. Here we have proportion. It's going to bind to the receptor. The leptin, that binding indicates to the eukaryotic cell to produce more leptin. As the production of leptin goes up, that is actually your manufacturer distribution area, which is your Golgi rough smooth ER. It's going to ship them out in vesiculas right here through the membrane. Those are signals that there's enough fat in the body to do the appetite. Here's your scale. Again, this is really comes down to structure and function. The bigger things are, the more function they have, and kind of went over this stuff. Thank you very much.